All right, let's dive into one of the most incredible stories happening inside you right now, protein synthesis. This is basically the instruction manual for how your body takes the code written in your DNA and uses it to build, well, you.
You know, we hear it all the time. Your DNA is the blueprint for your life. It decides everything from your hair color to how your muscles work. But did you ever just stop and think, how does a code, a sequence of letters, actually build something real? How does it make stuff?
Well, the answer is this amazing two-step mission called protein synthesis. It's a process that starts with the master plans in your DNA, makes a copy in a step called transcription, and then uses that copy to assemble the final product in a step called translation. We're going to walk through this whole mission right now. So, every good mission starts with a problem, right? And our cells have a big one. The master blueprint, your DNA, is way too valuable to risk damaging. So it's kept on permanent lockdown inside this high-security vault called the nucleus. But the construction sites, the little factories that build proteins called ribosomes, they're all outside the nucleus. So check this out. You've got the DNA, the master plan, stuck inside the nucleus. It's just too big and important to ever leave. And on the other side, you've got the ribosomes, the protein factories, hanging out in the cytoplasm. The factories can't get to the blueprint. So what do? You need a messenger.
And that brings us to the first part of the mission, transcription. The goal here is pretty simple, but brilliant. The cell needs to make a temporary, portable copy of just one specific instruction from that massive DNA blueprint. A copy that can actually get out of the nucleus and take the message to the factory. And the hero of this first act is a molecule called mRNA, or messenger RNA. The best way to think about it is like making a quick photocopy of a single recipe out of a giant, ancient cookbook. It's small, it's a single strand, and it's built to be the perfect getaway vehicle to sneak the plans out of the nucleus.
So how is this copy made? There's this incredible enzyme called RNA polymerase that's basically a master scribe. First, it finds the exact gene it needs to copy and latches on. Then it unzips a tiny section of the DNA double helix. As it scoots along one of the strands, it reads the code and builds a matching mRNA copy, base by base. Once it gets to the end of the gene, boom, the freshly made mRNA copy is released and is on its way out of the nucleus. Now, here's a fun little quirk. RNA speaks a slightly different dialect than DNA. When the copy is being made, C always pairs with G, and T on the DNA pairs with A on the RNA. But here's the twist. DNA's adenine, or A, pairs with a base called uracil, or U, in the mRNA copy. It doesn't use thymine, or T. This little switcheroo is the secret signature of RNA. Okay, phase 1 is complete. Our messenger has escaped the nucleus with the secret plans. Now the mission moves into its second and final phase, translation. This is where the action happens. It's where that abstract code is finally read and translated into a real physical thing. So our little mRNA molecule makes its journey through the cytoplasm until it finds what it's been looking for, a ribosome. This ribosome is our protein-building factory. It immediately clamps onto the mRNA strand, ready to read the instructions that were just delivered.
Now the ribosome is super efficient. It doesn't read the code one letter at a time. It reads it in 3-letter words called codons. And each codon is a very specific instruction for one of the 20 amino acids, the building blocks of proteins. So for example, the codon AUG is the start signal, and something like UGG always means add the amino acid tryptophan.
But how do those amino acids actually get to the factory? Ah, that's where another hero comes in, tRNA, or transfer RNA. Think of tRNA as a fleet of specialized delivery trucks. Each tRNA truck is designed to recognize just one specific codon on the mRNA, and it only carries the one specific amino acid that matches it. So here's the assembly line in action. The ribosome is locked onto the mRNA. The first tRNA truck pulls up, its code perfectly matching the first codon, and drops off its amino acid. Then the ribosome slides down one spot to the next codon. The next correct tRNA zips in, drops off its cargo, and the ribosome forges a chemical bonds between the two amino acids, starting a chain. The first tRNA leaves, empty, and this just keeps repeating shift, deliver, bond, release, over and over at incredible speed, building that protein chain one piece at a time.
The ribosome chugs along the entire length of the mRNA strand until it hits a stop signal. And just like that, its job is done. Mission accomplished. Almost. We have this long chain of amino acids, but it's not quite a protein yet.
What we have at this stage is a complete polypeptide chain. It's basically a long string of beads where each bead is an amino acid assembled in the exact order that was written in the original DNA blueprint. The plan was followed to perfection.
And now for the grand finale. This is where the magic happens. As soon as it's released, that chain of amino acids begins to twist and fold in on itself, creating an incredibly complex and unique 3D shape. And that shape is everything. That final, folded structure is what gives the protein its power and lets it do its job, whether that's digesting your food, contracting a muscle, or carrying oxygen in your blood.
Now, just let that sink in for a moment. This entire mission from a gene locked in your DNA to a fully folded working protein is happening inside your body trillions upon trillions of times, every single second. It is constantly building and rebuilding you from the ground up. So the only real question is, what will you build to?